That's us live. 3, 2, 1. We've done that bit. Good afternoon, good evening, good morning, everyone. One of the beauties of these online seminars is that we can obviously get participants from all across the world. So welcome from wherever you are. We won't start straight away because there's always a few people that are, just logging on as we sort of start these things. We'll give it a few more minutes. But just to make sure for the people who have logged in already, you're in the right place. And yeah, we'll just give it a few more minutes till people turn up. And I guess I can use this to give a little bit of housekeeping, which I'll repeat again when we do really start. But for any questions, please put them in the chat column, which is on the right of your screen. There will be potential sort of questions or any, interactions that you may want with your fellow attendees. If some of you maybe even have some information to add to either the talks or to the questions as they come in, we'll try and address as many of those questions as we can. What we tend to prefer to do is if you can maybe wait till the end, and then that way we can put any questions to either Will or to Cadler or to both at the end. And if that is a case of you want it to a specific person and maybe even a specific slide, please keep a note of the slide number and maybe also the potential presenter that you would prefer it to be questioned to. We'll try and get through as many of those as possible. We also do have, beside the chat button, you've got the polls, which we've got 3 questions for you to potentially sort of click on. Very simple questions around sort of the topic in hand. So please do take a look at those polls. And also just right of the polls button is a handouts, which is also quite a useful feature. We've got a number of PDFs there for you, whether it be the review paper that Will, Cadler and myself and some colleagues wrote together on iPSC and APC developments recently, or whether it be application reports upon some of the developments that have happened on iPSC and APC capabilities. Unfortunately for the people that have heard that, I'll probably be repeating most of that soon. But we are at quite a good number already. I don't want to spend too much time, but just a quick review for those who may have turned up late. There's a chat button. Please put any questions in the chat. There's a polls button for questions regarding the theme and the topic of choice. And there's a handouts button for any PDFs relating to this theme. I will start the introductions now, I think, because we're probably a good sort of time spell and we've got a good number of people online already. So once again, welcome everyone to this webinar. This is our third in a series of automated patch clamp and iPSCs. The previous 2 were with Liz Buttermore of Boston's Children's Hospital and Mike Hendrickson of BrainZell. They were both on neuronal derived iPSCs. Today's is still on iPSCs, but we're now looking at cardiomyocytes derived from iPSCs. And there's been some great work from Niels Voigt lab in Göttingen, and Will Seibertz is one of our speakers from that lab, and he'll be telling you about some of the great developments they've had in terms of maturation and induction of iPSCs. And then that will be followed by my colleague Cadler, who has been taking some of these great iPSC-derived cardiomyocytes from the Voigt lab and doing some APC recordings upon them. I don't want to take too much more of your time. I'd rather the speakers get the stage and speak for themselves and give you their great sort of data and work. So without further ado, I'll hand over to Will. Over to you, Will.
Great, thanks Damien for that introduction. I will immediately start sharing my screen and get this on the road. So hopefully you can all see my screen there. And yeah, as Damien briefly said, I'm a PhD student in the lab of Professor Niels Voigt. And I'll be doing the first part of this talk today, focusing on induced pluripotent stem cell-derived cardiomyocytes. Sort of what are they? How can they be made in a simplified way? And of course, looking at various ways in which maturation can severely impact their function as we go. And of course, Catalina will take over the second part. So without further ado, we've got here a human heart during open heart surgery. Now, in order for the heart to beat in an organized way, the cells of the heart, or cardiomyocytes, as we call them, must possess the ability to conduct ions across their cellular membrane. This enables this activation of the cell and then the subsequent contraction that we can see here with the beating heart. Synchronized ionic movement through a variety of proteins within the cell membrane, these are these cartoon colorful things shown here on the right, actually enable the generation of a voltage difference across the cell as these charged particles move from inside to outside, or vice versa. During an activation event, these precise ionic movements actually create a transient reversal of this membrane voltage, as it were, which is known as an action potential. And if we trace the voltage change during this event, we can see a trace that's shown here now on the right, where we get a sharp kind of peak and a gradual reduction down to resting levels. So this is all basically generated through a unique synchronized dance, as it were, of all of these different ions moving in and out of the cellular membrane in a time-dependent manner. Once we get this activation event, this then leads to the contraction of the cell, and then globally, of course, to the contraction and the beating of the entire heart. So these, of course, are very important for the function of cardiovascular health in general. So all these specific channels and proteins in the membrane need to be functioning very well in order to sustain life, as it were, in a human or in a mammal. And therefore, when we study them, when we make models of these cardiomyocytes within a lab, they, of course, need to replicate this physiological system as closely as possible. And one such model that we do use to try and replicate the cardiomyocytes of the heart is induced pluripotent stem cell-derived technology. And this is very nice because this essentially allows us to create an infinite amount of cardiomyocytes in a lab without having to rely on, for example, animal experiments or patient biopsies from open heart surgery. So many in the audience are probably familiar with this kind of technology, so I'll keep my overview relatively brief, but hopefully still interesting for those listeners out there. Firstly, these stem cells are essentially created from somatic cells from patients. What that means is we're actually able to take fibroblasts from skin samples or various cells within the blood from a standard blood test, and we're able to transduce various transcription factors using either viruses or other technology. We can transduce these 4 so-called Yamanaka factors based after their founder or the person who found these unique cocktail of 4 factors that actually cause a shift in the age of a cell that can essentially put them back in time to a pre-embryologic state. So that means we can take a normal cell from a human, and then we can essentially shift that cell or de-age that cell to this pre-embryologic state. The cells that are then created after this process are called induced pluripotent stem cells, or iPSCs, which you'll be hearing a lot about. Now, an interesting little fact, when we look at the process of human embryo development, and we talk about these iPSCs being pre-embryologic, what we're actually talking about are these cells here shown in this little picture in the lower right. These correspond to the inner cell mass of the developing blastocyst about 5 days after oocyte fertilization. This means that these cells are pluripotent, meaning they can change into any of the main 3 germ layers that make up a mammal, as it were, or and not any extra embryologic aspects such as the placenta or the trophoblast. So what we're looking at here are the cells that can essentially form any part of the human body. We are able to create those in a dish, And of course, we're going to be talking about heart cells and cardiomyocytes. So I'd like to give a brief overview of what we do in the lab while we have these cells in a dish, how we can push them to become cardiomyocytes. And the first thing we do is activate a particular cell signaling process known as Wnt signaling. After 2 days of activating this particular cellular process, which can influence the nucleus to create a variety of different proteins, which can then force the cell into this mesodermal fate, as we call it. This is essentially equivalent to around day 15 of a developing human embryo, where we get the formation of these so-called germ layers, which end up forming a human and all of their organs, or organs within a mammal. And you can see here the red cells here indicating the mesoderm formation. After only 2 days of activating the signaling pathway, we can get quite a nice population of mesoderm cells within our Petri dish. Now, moving on, we want to then stimulate these mesoderm cells to be specifically orientated to become heart cells or cardiomyocytes. And what we do then is we simply just inactivate this signaling pathway. We block various proteins within this cell signaling cascade and inhibit the entire pathway. By day 8 of culture within a dish, we can therefore assume that we have a very nice mixed population, some cardiomyocytes and some other kind of messy contamination cells that haven't quite got the message yet. So here a little overview. The Wnt signaling cessation or stopping promotes a cardiac phenotype. And this is loosely equivalent in the developing embryo to when the heart fields start to form and the heart organ starts to form as well, roughly around day 20 in a developing embryo. For us in the lab, we actually already see the beginning of cellular contractions within the Petri dish already at day 8. The last step here is selection or purification, where we actually quite simply just remove any contaminated, any non-cardiomyocyte contaminants within our cellular culture. So we see here we go from a mixed mesoderm, we don't really know what we have. And what we do is we remove glucose from the environment that the cells are sitting in, from their medium, essentially. We remove glucose and we introduce lactate, meaning we force the cells to use lactate oxidative metabolism as a main way to generate energy. Fetal cardiomyocytes are very happy with this, so they're happily pumping away. However, any non-cardiomyocytes, they will not survive this process. So that means theoretically, after this 20-day process, we can now assume that we have a relatively pure population of induced pluripotent stem cell derived cardiomyocytes, or iPSC-CM, which you'll see an acronym regularly used throughout the talks. Here's a nice picture where we've stained the cardiac specific muscle fibers green. This in particular is referring to alpha-actinin, only found in heart cells. And when we stain for this particular protein within our iPS cell cardiomyocytes, we can really see a nice overview of many alpha-actinin positive cells. And we can even see that striated or stripy pattern across these cells, indicating that they do actually have very nicely formed muscle fibers already, which allow them to contract. The data here was from flow cytometry, and this is just from a representative batch of our cells that we create in the lab. This is shown around 90% pure for alpha-actinin, indicating we have 90% and above pure populations of iPS cell cardiomyocytes. Here's a little video of what they actually look like under the microscope. And you can really see, although it's a little bit flustered and a little bit busy, you can really see this really regular contraction in each cell within this dish here. So we really assume these to be very nice, pure cardiomyocyte populations. Once we leave these to sit in cell culture for a little while, we begin to see some form of passive maturation processes. We see a slightly more elongated cell shape, and we really see really nice, slow but strong contractions as that cell beats, just like a heart cell would, in this case, in a developing fetus. So we see a contraction in these cells, which means we essentially have some form of correct ionic signaling or function within these cells. We do or this indicates essentially that we are experiencing these action potential events that I referred to before. However, you'll see here on this figure that the action potential shape for these iPS cell cardiomyocytes is very different compared to the native CM or the adult cardiomyocyte situation. And this is because the distribution and the conductivity of the ionic channels within the membranes of these iPS cells are not yet at a level where they can exactly replicate that of the adult cell. So you'll see in all of these colorful current diagrams below the action potential, most of them are smaller or indeed non-existent in the iPS cells. And this creates, in the current literature, the current way of thinking, a much more shortened and a smaller overall action potential shape in these iPS cells. A crucial part of the current knowledge in this field in developing these iPS cardiomyocytes is the perceived complete lack of a specific ionic channel which conducts potassium ions. In this case, this is the basal inward rectifier current, also known as IK1. As you can see in the human situation, or in the native cardiomyocyte situation, this current is very active, pumping potassium ions out of the cell at the end of the action potential. So when we see that nice shoulder coming down to resting values, usually we expect this current to be active. However, the current way of thinking in this field is that these IK1 channels are essentially completely absent in these iPS cell constructs. And that's, of course, a problem because we want to be mimicking an adult phenotype, as it were, exactly as close as we can. So I'm going to be giving a little bit of our data about discussing this perceived lack of IK1 and how maturation processes can actually reveal this to be not quite true. This idea of maturing these cells is also very much a current topic in this field. How can we make these cells also functionally, of course, but also structurally and metabolically represent an adult cardiomyocyte? So we've got many techniques while these cells are in culture that can attempt to push these cells toward a more mature phenotype, as it were. These involve the use of chemical stimulants to try and essentially use drug cocktails to enforce some kind of mature phenotype in these cells. A particular method that's becoming increasingly popular is the use of tissue engineering to generate three-dimensional models of these cells. These tissues exist as a complete structure. They do have cardiomyocytes embedded within a three-dimensional situation, which more closely replicates, of course, the situation in an embryo or in a human, where, of course, you have an organ that works together, not just a flat layer of cells in a dish. Cold culture has also been used. Of course, it's common knowledge that the human heart does not only contain heart cells, it's got a variety of other types, fibroblasts, etc., that can perhaps induce some paracrine or hormonal activity that can influence the cells to become more mature. And of course, maybe not so flashy, but still important aspect of how these cells can become mature is simply by leaving them sitting in their dish and seeing what happens after a long time. This long-term culture or passive maturation is what I'll be looking at today. It's been not so well standardized within this field about the ages at which iPS cell cardiomyocytes are measured, for example, for their functional characteristics, for their ionic currents, the differentiation processes, and the time at which they are measured does show quite a lot of variation. So this is something we were interested to look at in our lab. I'm going to be discussing data using manual patch clamp, whereby we can approach these cells with very small glass pipettes. And once we form a connection with the cell, we can break into the membrane and essentially acquire an electrical connection with the inside of these cells. Once we have this electrical connection, we are then able to isolate a specific current, which is carried, of course, by those ionic channels that I discussed before. And then we're able to measure the current itself. We apply a voltage protocol, which allows us to select exactly what we want to be measuring. And then we are able to acquire the ionic currents from these cells based on the current of interest, whatever the experimenter wants to measure. In this example here, this is a calcium current, an L-type calcium current that I'm showing here. This L-type calcium current is the first thing that we looked at in these cells in terms of their maturation. And we did see, of course, that we did see sort of robust L-type calcium currents in these cells. I'm showing here on the left a representative trace from an early iPS cell, early meaning somewhere between the age of 30 days and 50 days. And we can see a nice current there. And we can also see a calcium transient below it. This is a simultaneously triggered transient, which represents the systolic calcium release during an activation event. Not so important for our focus today. But what is interesting is that when these cells get older and they're just sitting in their dish, they actually change these characteristics quite significantly. We see an increase in the L-type calcium current amplitude, and we also see an increase in the amplitude of this calcium transient as well. Looking at the data points here, orange being older and blue being younger, we can see a significant increase in both the L-type calcium current density and the amplitude of this transient, simply by waiting for these cells to become older in their dish. If we look at this progression on a day-to-day basis, we can really see a clear linear relationship here. As the cells get older, the L-type calcium current amplitude, or sorry, in this case, density, does tend to increase. And that was very interesting for us because that opened up questions about what other ionic currents also increase after long-term culture that might be easily missed if these cells are measured at younger days. And so, of course, we went back to the IK1 question. As I said before, most people assume that IK1 is very minimal or basically absent in these cells. What we first wanted to quickly check is if we can detect the presence of the protein that conducts the IK1 current within these cells. So we did a little Western blot here to quantify the amount of protein that we could actually see, or the amount of protein that's expressed. And we saw a significantly increased amount of the CUR2.1 protein that conducts the IK1 current in later cells or older cells compared to young. And so this was our first kind of surprise, as it were, in the lab. These cells seem to have the protein. Do they actually express the current? Can we measure the current itself? And so what we did is we applied the voltage protocol. And in this case, we applied a specialized ramp protocol in the presence of specifically designed bath solutions in order to isolate exactly the current of interest that we want to measure. When we apply a depolarizing ramp protocol like this, we're essentially depolarizing the cell and looking at the relationship between the current and the voltage as the voltage changes. So it's essentially a real-time IV curve, which I'm about to show here. And as you can see here on this data, when we look at the inward component of IK1 measured at minus 100 millivolts, indicated by the arrows here, in the early iPS cell cardiomyocytes, we detect some, but very small amounts of IK1. However, this is significantly increased as the cells get older. And this representative trace here shows from a late or older iPS cell cardiomyocyte, really clear increases in IK1 current density. Once we look at that in a sequential way as well, we can see a clear linear relationship here. As the cells seem to get older, these parameters change, and IK1 does tend to increase in these cells. And so that was very also, interesting for us to see. And we wanted to, of course, keep on investigating this phenomena. And what we did is we measured a variety of different currents and parameters within these cells, both as they were young and as they got older. I won't go through any more of the specific data, but what we did here is we compiled all of our information that we'd found as the cells get older, and we combined it with an already established computer model of a cardiomyocyte. And it's shown in this publication here from Koenig et al. From 2019. This computer model essentially creates an in silico cell, which means that this simulation of a cell contains as accurately as possible every single electrical or functional process that happens in a normal cardiomyocyte should also be happening in the computer model. Therefore, we can manually change a specific parameter in the model, and we can see what happens essentially as if this cell was a real cell, as if we were giving a patient a drug, for example. So these models are, or this particular model is one of the leaders in the field. And that's also open source, which is very, very useful for labs like us, because what we were able to do is actually integrate our maturity-based data with the existing in silico data that has already been curated and prepared by this group. And we're actually able to then study the impact of maturity on the entire cell in silico, as it were. So what that means is, if I recapitulate here, these are our IK1 traces from early and late. When those are integrated into this in silico maturity model, we are then able to ask the computer, as it were, show me the IK1 from a day 30 cell and from a day 70 cell, and using its already established database of all of these different functions within the cell, it's able to generate a simulation here of what we expect to see. And this was also nice because this isn't just replicating our data and making it digital. This is using an established format that has already been created. So to already be able to see that the integration of our data can produce a maturation-dependent increase in IK1 was very satisfying for us to see. And we felt that now we have quite a powerful model with which we can assess any aspect of cellular electrophysiology within these iPS cells using this maturity-based in silico model. And that's essentially exactly what we did. We said, okay, then now we've got this nice maturity-based model. What happens to the action potential as the cells get older? So we've got all this nice dance and this concert of different changes in ionic function as the cells get older. What happens to the action potential? Shown here in blue is the simulated action potential when the cells are 30 days old. And then as I skip through here, the cell gets progressively older, and you'll begin to notice by day 50, the action potential also gets progressively shorter as we go. And that was also quite surprising for us to see. Oftentimes the predominant way of thinking about maturity is bigger, better, make all the currents bigger, make the action potentials bigger. But what we're seeing here is we started big and we actually get a shortening of that action potential duration as we go. It's shown here in the graphs below as well. So we see, firstly, like I discussed, we see some increases in some currents like IK1. We see no changes in other currents. Why do we see a shortening of the action potential duration? We ask, could this unexpected result of the IK1 increase play a role in the maturation-dependent action potential shortening that we observe? So here, using iPS cell cardiomyocytes that have been loaded with a voltage-sensitive dye, we're able to see a nice trace of these action potentials as they are elicited within a cell. Here we've got one from an early iPS cell cardiomyocyte, and here from a late. And here already we can see nicely that our in silico data from the computer model is replicated here experimentally. We do see a shortened action potential duration in these older cardiomyocyte populations. When we block IK1, when we remove it completely from the cell by blocking it with barium chloride, we also see some interesting results. We see no change in the young cells. However, we see a prolongation of that action potential in the older cells when IK1 is removed or blocked, essentially. This prolongation was significant in our cellular cohort that I'm showing here. And so that was very interesting for us because that essentially answered our question that IK1 does appear as the cell gets older. The increasing amount of IK1 expressed in the cell appears to play a large role in maturation-dependent action potential shortening. Now, in the literature, many people associate IK1 with the maintenance of a stable resting membrane potential. When the cell is resting, when it's not experiencing action potentials, the voltage is kept constant at a very negative value. Usually, IK1 is assumed to play roles in keeping that steady voltage state. And so what we saw here was when we applied the barium chloride to the young cells, we saw no change in the resting state between action potentials in the young cells. However, as you'll see clearly here in the late cells, we began to see very much arrhythmic activity here as the membrane became less stable. So that was really nice as well, and quite convincing for us, and hopefully for the audience as well, that IK1 does appear to be quite a major player in this maturation dependent shortening that we see in the action potential. Now I'd like to backtrack a little bit, because yes, IK1 is very important for the maintenance of resting membrane potential. Therefore, why did nothing happen in the model? So this is the same slide that I showed before. We see the action potential getting shorter, yes. But when we track the progression of the resting membrane potential, we essentially see very little or no change in the resting voltage there. And that was quite surprising, because we would really expect to see more IK1. We expect to see a more negative resting membrane potential. So we wanted to investigate this further. We applied a different method to investigate action potentials, again using manual patch clamp, and this time using the current clamp configuration. Again, here we see a general shortening of in these representative traces. We see a shortened action potential in the late cohort. However, when we assess the resting membrane potential, we can also see, similar to what our model said, that in our experimental data shown by the dots, the blue and the orange dots, we see no change in the resting membrane potential. And that, so that, of course, gave us something to be curious about, and we wanted to find out why. We went back to our model, and that's, of course, again, the advantage of having such a nice computerized platform, because we can manipulate anything we want. So we went back to it, and we essentially removed IK1 from the cell, but we kept the cell functioning normally. So it grows up as it normally should. However, this IK1 increase is removed. And as the cell gets older, as I click through, we can see a progressive increase of the action potential duration, which goes in line with our findings. When we do have IK1, it gets shorter, remove IK1, now it's longer. And eventually, as the cell gets older, we see the generation of these triggered events and this arrhythmic activity within the cell. So this all follows normal thinking, or expected thinking based on our hypothesis about IK1. But it doesn't quite explain the resting membrane potential, because here, without IK1 involved, we see a progressive increase. You'll notice that here the units are flipped. So if it goes down, that means an increase in the resting membrane potential. As the cell gets older, resting membrane potential increases. And we wondered, why is that? That means there must be some other current which is acting in the background, which is counterbalancing this IK1 mediated typical decrease of resting membrane potential. And so we simply went back to the schematic here, and we looked at which currents are active in the end or at the resting phase of these action potentials. And we identified, of course, the NCX exchanger, or the sodium calcium exchanger. This is in fact active at the end of in the end phase of the action potential, phase 3 and phase 4. And based on our maturity-based data, which I have not shown so far, we also see an increase in this current as the cells get older. So we wondered, okay, could that current then be responsible for this increase in resting membrane potential? And indeed, what I'm showing here already is when we block this current, this NCX current, we in fact see, predominantly here, I'll direct your attention to the resting membrane potential, we see a sharp decrease of resting membrane potential pretty much back to the resting values at day 30. And what that told us was that we have identified our culprit, as it were. Our little suspect, NCX, is the key. We do have maturation-dependent increases in IK1 as the cells get older. However, the changes in resting membrane potential are counterbalanced by the development of progressively increasing sodium-calcium exchanges within the membrane as well. So with that, I'd like to do a little bit of advertisement here. All this work will be contributing to a software platform and a publication that will be released very soon, we wanted to generate a new interface that would be freely available, whereby someone can essentially input the exact age of their iPS cells or their iPS cardiomyocytes that they may have in culture, input the age here, change any of these ionic currents that the experimenter may want to test, whether it's blocking, whether it's using an agonist, whatever it is. And then the output will be a real-time simulation of what you can expect based on the age of the cell and your specific modifications to the ion channels in the model as well. So this is called the immature software. Here, I for iPS cell cardiomyocytes. And we hope to have this released very soon, especially by the end of the year. This should be freely available. And so definitely encourage any interested listeners to stay tuned, and the software will be freely available very soon. That's essentially the end of my part here. As a quick wrap up, I'll go over that indeed such a simple maturation technique, just like leaving the cells to passively become older, really does show very complex temporal dynamics in how these currents develop and mature. Secondly, we did show that IK1 is definitely present in these iPS cells, and that one depends on the age of the cells, or how easily it can be measured, and two, that it definitely seems to be important for action potential morphology. And overall, we believe that this idea of keeping the cell age in mind is very important to consider during various disease modeling projects or drug development strategies. So I'd like to thank you all very much for your attention from my part. I'd of course like to thank my supervisor, Professor Niels Vogt, as well as Ayste Lutkutte for her support with the cells and cell culture in general and calcium measurements. Melanie Ritter as well, who helped with the action potential measurements, and Rebecca Dolk as well from our lab. Special thanks, of course, for Sophion for allowing me to present our data for you today. And as Damien said, I believe questions for both of these talks will be had at the end of Catalyst talk. So with that, I'd like to hand back over. And yeah, again, thank you very much for your attention. Cheers.
Fantastic, Will. Thanks very much. That was really nice, and such a nice overview and great data you've got there. As you say, we'll take the questions at the end. Keep them coming in on the chat. I will pass over to Cadla straight away because I know she's got some lovely data and information to impart to you all. I will be back soon, and we'll have the questions at the end. Till then, take care.
Thank you, Damien. I'll just share my screen. I hope you all see it. Otherwise, you have to say something because I cannot see your faces right now. In any case, my name is Katla, and I'm working as a research scientist at Sophion, and today I will present the preliminary results of a project that we were actually running last year, where we aimed at developing automated patch clamp assays for the electrophysiological characterization of iPSC-derived cardiomyocytes. So Will already introduced very nicely the iPSCs. So the specialized cells that you can derive from these induced stem cells were originally thought to be used primarily for regenerating damaged or diseased tissue. But since then they have been considered for a range of applications where we think automated patch clamp could be a good high throughput tool. For example, now we have the technology has given access to large quantities of these specialized cells, but there's still a requirement for high throughput characterization and quality control. We can derive these cells from a patient, which means that you can do disease modeling, you can do drug screening and discovery, pharmacology and safety screening, and maybe even at some point personalized medicine. So the main vision of this project that we were running last year was to develop automated patch clamp assays for characterization and screening of iPSC-derived cardiomyocytes. And I will just briefly go through the main challenges that we are facing when we are trying to do these measurements, how we try to address them, and then a few proof of concept applications that we have developed. So what we have experienced these last 5 years that we have worked with iPSC-derived cell lines and specifically cardiomyocytes is one big challenge is within the differentiation and maturation protocols. And we are actually not really involved here. We are working together with cell line providers, collaborators that are doing the differentiation, and then we might take part a little bit in the maturation process. But what we experience is that there is still a requirement for quality control and standardization. We see a large variation between cell lines from different vendors, and even within the same or within batches that comes from the same cell source. We also see sometimes that the cell population that we are looking at is very heterogeneous. We have the cell line that we are looking for, and then we also have other contaminants within. And then, as Will also talked about, what we often observe with iPSC cardiomyocytes, or what is a general problem, is that the cells are immature as compared to their primary counterparts. So as I said, we're not directly involved in the differentiation, but the way we address this is that we have established close collaborations with iPSC cell line providers, which mean that we can measure the cells, and every time we see something that we don't understand, we can get back and discuss what happened, how was the differentiation going, and we can also maybe give some feedback on quality criteria and thresholds and so on. The second challenge that I listed here is one we have worked with a lot in-house. So this is due to the fact that, at least until now, you have to add the cells in suspension to the APC machines. So especially for cardiomyocytes that like to form these dense tissue-like cell cultures, as you see here, the lifting of them from the surface and the dissociation of them from each other causes a lot of damage to the cells. That means in the cell suspension, you risk having a lot of dead or dying cells. You might have debris and cell aggregates. So here we have worked with the harvest conditions to identify the best protocol to ensure both you want to have an efficient but also gentle dissociation. Then we have worked with purifying the cell suspension. You can remove debris, dead or dying cells. You can remove aggregates. And then finally, we have also worked with sorting of the cells based on structural and functional markers, so that you work only with the cell type that you're interested in. So, as I briefly mentioned, even though we are not really involved in the differentiation of the cells, or we are not doing that ourselves, we have been involved in the maturation of the cells. So typically we receive the cell lines at an early stage, where they're still a bit immature, and then we keep them in culture in 1 to 3 weeks for them to mature further into the specialized cell that we're interested in. And as Will already mentioned, there are many different strategies to increase the maturity of the cells. And we work together with the cell vendors and our collaborators to apply these different maturation strategies. And what we have done, for example, is to measure then how do the ion channel expression look at different time points, or how does it look when you have them in when you culture them in different types of maturation media, and so on. For the harvest, we have tried a lot of different things. We have I think by now we have a good starting point. We have some protocols that we always start out with, and then based on how the cells look, we optimize those further. we still see, and I think maybe this will always be the case, that there are so large differences from cell line to cell line of the iPSC cardiomyocytes that we have to optimize the harvest individually. One thing you can do, for example, if you have this very dense tissue like cell culture, and you see that a lot of the cells are dying or damaged during the harvest, is that you can replate the cells. You can replate them at a lower density. That allow you, and you can do that maybe 24 hours before your experiment, and that allows you to remove all the dead cells. The ones that survive have some time to recover, and they can then also, if they're plated out in a lower density, they don't get the chance to form these very tight connections, so that you can use a more gentle dissociation method in the second step before your experiment. And of course, there's a lot of different steps that you can apply here that I will not really go into, but many of these has to be optimized every single time we're working with a new cell line. Then it's different again from every cell type we work with, but often we see that we end up with a cell suspension like this, even after optimizing the harvest conditions. That you have your target cell, here the iPSC cardiomyocyte, but you also have aggregates, you have some debris, you have some dead or dying cell, and you might even have other cell types in there that you're not interested in. So for this specific cell line, when we applied the cell suspension directly on our machines, we got a success rate of about 10%. Based on our quality filters. These are membrane resistance and capacitance measurements, and also filtered on sodium current. So just adding another step where we incubate with DNAs to degrade DNA and make the suspension less sticky, and we filter to remove aggregates, increase the success rate by a factor of 2. And then sorting this cell suspension for a positive marker, so that we get rid of both debris, but also the cell line that we're not interested in, or the cell type that we're not interested in, increase the success rate by a factor of 2 once again. So for this last step, it's important to mention that you need to work with a cell sorter that is very gentle on the cells, because we want to be able to measure on them directly afterwards. We tried different cell sorters, and what worked for us was this Milteny Taito cell sorter. Another thing to mention is that you need to have a large starting material, large quantity of cells to start with, since you do lose a lot of cells in the process. Here we ended up selecting 20 to 40% of the cells that were in our starting material. Once you have a good cell suspension, we can start doing our measurements, and this is the first proof of concept application that I will show you today, which is the one that we have been doing most and has been working with most, which is characterization of the cardiac voltage-gated ion channels. So here you see a plate view from half a Q plate, so it's 24 sites. The green here are the cells that passes the quality filters, and the red are the ones that fail. And here you see the sodium currents. So these measurements that I'll show you now was performed on cells that were provided by Will and his colleagues in the laboratory of Niels Voigt in Göttingen. And in this cell line, we saw that out of the cells that passed quality criteria, 90% of those we measured sodium currents. You see here an example of a sodium current and the current voltage relationship. Another ion channel that there's also a big interest in, and that Will also mentioned, this is CAV 1.2 channel, we have in the cells that we received from Will, we see 80%. In 80% of the cells that pass quality criteria, we see the calcium current. You can see here the current in black before and red after addition of nifedipine, a CAV 1.2 blocker. And you can also see here the current voltage relationship as an average of 28 cells before and after the blocker. Then an ion channel that is very interesting, especially for cardiac safety, is the HERC ion channel. The HERC current is in general very small. That is the case for primary cardiomyocytes as well as iPSC cardiomyocytes. In this cell line, we were able to measure HERC currents in about 60% of the cells that passed quality criteria. The strategy we used, because the current is very small, the strategy we used to measure these currents was to increase the extracellular potassium from 5 millimolar to 75 millimolar, thereby increasing this tail current. You can see in gray is 5 millimolar. In black is then this increased tail current measured in 75 millimolar potassium that we could subsequently block by 0.5 micromolar of E4031. And here on the right, you see the current voltage relationship before in black and after in red, addition of the blocker. Finally, we could also measure in this cell line the IK1 current in a little bit less than 60% of the cells. We use the same strategy as for the HERC channel. We increase extracellular potassium to increase this inward potassium current. And then we blocked using 1 millimolar of barium. And then we use a feature of the analyzer that has not been released yet, but will be shortly, where we can subtract those currents from each other to plot only the barium-sensitive current, which is then the KIR2-1 current, and you can quantify the current voltage relationship as shown here on the right. So for this specific cell line, we see in 90% of the cells, we can measure sodium currents, 80% we can measure calcium, around 60% we can measure HERC, and a little bit less we can measure this IK1 current. And when we then go on to look at action potentials, we can see that for the cells that fire action potentials, they need all of these ion channels to be present, which makes sense, but is also also very good to know when we try to find out how large a success rate can we expect from our action potential measurements. So that kind of leads me to the final application, or the other application I want to show to you today, which is the measurements of action potentials. And again, Will already went through this. iPSC-derived cardiomyocytes are kind of known for having this immature shape of the action potential. They express some ion channels that they should not, and they are lacking some other ion channels in order, or when comparing them to their primary counterparts. But as you can also see from Will's presentation, the field is getting better and better at producing and understanding these iPSC-derived cardiomyocytes. And with the ones that we were working with from Will and his colleagues, we can measure action potentials, as I said before, in about 50% of the cells that pass quality criteria. And that corresponds to about 10 sites in a Q-plate. So 10 sites out of the 48 sites that we have in a Q-plate, we can measure action potentials with these cells. And you can see already that the shape of the action potential vary a lot from cell to cell. I don't think we really understand yet whether this can be optimized, whether we can get a more homogeneous phenotype for the cells, or whether this is the inherent variability that you would see from cells in a culture. But we can quantify the action potentials. We can get, so here we have extracted the threshold potential, the peak potential, the hyperpolarization potential, and the duration of the action potential at 90%. And what we can see is that the largest variation that we observe is in this duration of the action potential. What you can also see is, as Will also mentioned, that the membrane potential is a bit elevated as compared to what you would expect. This is something that we are that might be addressed by improving the maturity of the cells, but that we are also trying to address by developing our current clamp feature so that you can actually hold the cells individually at a current that corresponds to the membrane potential that you wish that they have before you initiate the action potentials. Once we have once that we can measure action potential, so when we have a cell on a site that can fire action potentials, we can also modulate those by adding compound. So here you have in black the action potential measured in saline. Then you have from light red to dark red increasing concentrations of the CRV 1.2 enhancer, BK8644. And you can see how the action potential is prolonged. You can plot then the APD90 as a function of concentration for a single cell and for an average here of 10 cells and extract the EC50 value. Then you can do the same with the CRV 1.2 blocker. You have here the action potential in black and increasing concentrations of nifedipine that shortens the action potential. Again, you can plot the APD90 versus concentration for a single cell and for an average of cells to extract the IC50 values. So just to summarize, I ended up here showing you two proof of concept applications of APC measurements of iPSC cardiomyocytes. The first one, the characterization of the cardiac ion channels, we're using already in collaboration with cell vendors and cell providers to evaluate the influence of maturation methods. The second one is now the measurements of action potentials. I think we're at a stage now where we know that we can do these measurements. We still need to increase the success rates to actually make a feasible assay. Success rates and reproducibility of the measurements, I should say. And the main challenges still, we are challenged a lot by the variability between different cell lines and also from batch to batch and sometimes even from vial to vial within the same batch of cells. But there are improvements happening all the time, and there's a large focus on this in the field, so we hope this improves. Then for generating homogeneous and reproducible cell suspensions, I think we have now a very nice toolbox for of things we can do and protocols that we can apply depending on how the cell suspension look of the specific cell line that we receive. And we also have now a very good process for troubleshooting and for improving the cell suspensions. Finally, we have ongoing development of the current clamp feature, as I briefly mentioned. And our hope is, of course, that APC together with iPSC-derived cardiomyocytes can be used in disease modeling, drug discovery, precision medicine, and cardiac safety. Finally, I just want to mention that in addition to cardiomyocytes, we have also worked a lot with iPSC neurons. We're also starting up working with primary cell lines, and we are always open for collaborations. If you have some difficult cell line that you want to see if you can measure using our systems, you can just reach out. And we have these master and PhD student travel and research grants, also early researcher, that you can apply for if you're interested. Thank you for your attention.
Great stuff, Cadler. I'm just hoping that Will join us here on the stage in the spotlight, that we can answer. There's a lot of questions. Apologies to those of you that have to dash off. We have run slightly to over time, but I think that's because Will and Cadler had so much great work to show you. So I hope you don't mind. And if you can stick around, we will try and answer as many of the dozen questions we've got so far. We may not be able to answer them all, but we'll do our best to go through them as best we can. So I'll shut up and we'll go to the first questions. So a number of these obviously are for you, Will, because you obviously sort of went off first. So the earliest ones that we're going to go to first. Ralph asks, how many CTNT positive cardiomyocytes do you have in your preparation after lactate selection?
Okay, so after lactate selection, yeah, for CTNT, we have actually more than alpha-actinin. We have 95% and above CTNT positive cells.
Okay. Liho did ask a question, but I think we've pretty much answered that, Liho. So I'm going to skip on to Anissa's question. And this is actually sort of relating to a couple of questions that sort of came in around the maturation. So Anissa was asking, for instance, the L-type calcium current maturation seems productive, but are there any bad effects, for instance, of long-term maturation on other types of currents? I mean, even for instance, what you showed in the L-type, I did notice that the very end time point, it almost seemed to drop off in level.
Yeah.
Do you have
You're exactly right. Yeah, of course. So our thinking about that is that although I showed in those experimental data a nice linear line going forever, it's not linear, it's non-linear. It will have a plateau at some point. When we integrate that with the model also shows us that we see a plateau around the day 70, day 80 region, which is at the end of that graph that I showed. I would expect that severe drop-off we showed in the experimental data is simply batch dependence. Like Cadler mentioned, we see a lot of heterogeneity in these cells. So we do expect to see a plateau in this maturation-based development. And I believe that drop-off is indicating that.
Yeah, I mean, and I guess as part of that, I mean, the next question is almost along those similar lines. Daniel was saying, beautiful to see the L-type calcium current density increase over time. Were there also, I guess, morphological or even cell size changes over time?
For my experiments here that I showed you, we actually did not see a significant change in cell size over these 80 days of time. We always tried to also assess cell size, both by optical methods, by measuring it, the area, and by looking at the cell capacitance when we patched them. Both of these parameters showed no change in cell size, essentially. And we believe that because, of course, predominant thinking is that you expect to see more elongated and nice shaped iPS cells. We are of the opinion that because we plate these cells freshly plated at low density at day 30 for these experiments, we assume that by the end of our measurement window, in this case it was day 80, we assume that's simply not enough time for these cells to become structurally mature, as it were. We saw a huge variety, of course. I could show you, if I wanted to, I could pick out the best photos and there'd be rod shapes everywhere. But naturally, that variety is always there.
I mean, just quickly on that, I mean, these kind of sort of durations you're talking about now, 70, 80 days, is that pretty much typical within your lab, or do you even sort of see yourself going beyond the 80-day mark?
For us, that is typical, yeah. I would say, at least because we've been wondering about this maturation question, this passive maturation question for a while. Around day 60, day 80 is usually where we would think, yeah, that's probably okay. Anytime after that, we tended to see the cells become quite unhappy on their cover slips. They need to be plated in special conditions to allow for patch clamp or manual patch clamp measurements. And so, yeah, just due to technical restrictions, the cells just don't like being on these cover slips for too long. We usually are limited by extending that window without changing our protocols.
And the next question from Kalai, and I think this is almost applicable, or at least two parts of it are applicable to probably both of you. So the first one is probably more to Will. Since the differentiation protocol used does not target subtype differentiation, does the mix of cardiomyocyte subtype like nodal, atrial, or ventricular contribute to differences in RNA current observed? So that's more on your side, I guess.
No, excellent question. Of course, without any subtype specific special procedure, we do expect to see a mixture of cell phenotypes in our culture. With the stem cell unit here in Göttingen, we use the same cell lines that they originally provided us. They have done a lot of work to quantify exactly the amount of atrial specific proteins within the standard differentiation protocols. And we do of course see some atrial and nodal cells within that population, but our numbers are very low, and we have about, I believe it was between 92 and 95% ventricular isoform myosin light chain positive cells within the ones that I'm showing you. That means that we're looking at a significantly large portion of these cells are ventricular, and of course 5% of those they are atrial or nodal as well. But the 95 target was a good one for us to be fairly certain that it's likely we're measuring ventricular cardiomyocytes.
And I guess almost a follow-on to that, which is partly to you, Will, but I think also to Catalyst, since she's done some of these nice action potential recordings. I mean, I'm guessing that at least some of the heterogeneity that maybe sort of Cadler was explaining at the very end of her talk in terms of the action potential duration at the 90% mark, maybe that is down to some of this heterogeneity that Kalai's question sort of alludes
absolutely.
to.
No, it's absolutely correct. There's a lot of work that looks into even kind of spontaneous shifting of subtypes within standard cultures. The cells might begin their time slightly more ventricular-like, they might end up more atrial-like, or vice versa. It's absolutely, I would say, absolutely certain that there is some contamination in there which adds to that heterogeneity.
Yeah, just to add, I think once things get a little bit more reproducible, and maybe we get the success rates a bit up, this APC could actually be a good way of starting to quantify these developments and compare between different batches and different ages and so on.
Yeah, I guess because you start to get the sufficient N numbers to have that statistical power to sort of say, at day 30, we have 30% ventricular, 30% atrial, and 60% or whatever it might be, or 40%. There's my maths gone wrong already. 40% nodal, for instance. If you're going to break it down into 3 major subtypes, but of course you may even have subpopulations within atrial, ventricular, and nodal, for instance. Okay, the next question from Pablo. Have you checked if the density of ancillary subunits of channels like, e.g. KCNE or ERG, also change with time?
Yeah, easy answer, no. Yeah, we tried to measure as many currents as we can to get major currents that contribute to action potential to get an overview of this maturation development. But for example, checking subunits, we haven't gone into that detail.
And I guess, almost to you, Cadler, as well, like with, some of that statistical power that you get with the higher throughput, maybe at some point you'd be able to almost get some kind of definition pharmacologically or biophysically that might start to tease apart some of these, I don't know, well, certainly ion channel differences, but also potentially eventually maybe even subunit, differences that you might be able to sort of tease apart with some kind of pharmacology or biophysics.
Yeah, I think on the in voltage clamp, we're getting there, at least for some of the ion channels. Yeah, definitely.
Okay. And even though I jumped Liho's question before, because I think we had answered it, Liho, it wasn't just ignoring you. He's now got another question, luckily. Was NCX inhibitor tried on these iPSCs?
Not experimentally, no. No, we've just got the in silico data for now.
Okay. Yasunari asks, have you ever measured the currents using commercially available iPSC cardiomyocytes, such as iCell? And again, to some extent, Cadler did answer that in the sense that there are a number of different, iPSC creators that you've used, as well as, the great cells from the University of Göttingen. There were a number of different creators that you've used, for instance, and maybe Göttingen have also tried different cell makers.
From our side in Göttingen, we tend to stick with our own in-house lines. We find the process of freezing cells can be makes them very difficult to patch or manually, as we do in the lab. We tend to stay in-house for that reason.
Did you try applying any of the maturation methods that you developed to commercially available cell lines and see if that changed things?
No, we haven't done that. No. We tried a few different lines in-house, which seemed to show similar behavior, but never commercially available. No.
Okay.
And Ralph asks, did you check the metabolism of the cardiomyocytes in long-term culture as question 1? And then almost as a quick follow-up, are they still glycolytic or have they switched to fatty acid metabolism?
That is an interesting question, and we have not done any assays to test for that. So I cannot comment on that. That is an interesting point.
It is, yeah. I mean, because his follow-up point to that, or almost his reasoning behind that, you might be able to sort of see the question is, obviously, some of that metabolism may then still go on to have something that you could measure electrophysiologically to give you a different phenotype due to that change in metabolism.
Absolutely. Yeah, no, I completely agree. And unfortunately for the data I presented today, we haven't got that far. But that's a very, very important point.
And Ralph's peppering you with questions now. So another one from Ralph. Did you try the dead cell removal kit from Miltenyi? This would obviously avoid the positive selection, which might affect the function of the cardiomyocytes. I guess that's to Cadler.
We have not for this specific data that I've showed you today, but we have tried that for other applications with mixed results, I would say. But yes, in general, it's a good idea to do something like that. It needs to be optimized, and it needs to be optimized for every single cell line that you're working with, and it can work.
And Nazreen has a question, which may be to both of you, because it could obviously apply to Will's manual patch recordings and Cadler's automated patch recordings. What range was our seal for these mature iPSC cardiomyocytes? And the follow-up to that was, would you expect sealing changes with increasing maturity?
Good question. I think for the APC, the measurements I showed you today was all done in physiological solutions. And I think the seal resistant cutoff was 200 megaohms. But if I remember correct, I think the majority is between 500 and 1500 megaohms. Sealing changes with increasing maturity. What we have seen sometimes with some cell lines is that the older they get, the poorer they seal. And I don't think we exactly know why this is, but a hypothesis is that it's to do with some extracellular matrix or something that they secrete more and more over time that is actually making the sealing more difficult. We don't see it with all cell lines. to be a problem.
And I mean, this might also be a point that you were coming to, Will, earlier when we talked about that drop off in sort of some of the channel populations. I mean, potentially, certainly at present sort of standards, which seem to have taken off completely in the last year or so, and particularly with some of your work at Göttingen, but maybe there really is just a time point beyond which the quality, the health of the cell population is just starting to degrade, which would obviously then go on to affect potential recording metrics like the seal, the resistance on the seal.
Yeah, absolutely. Yeah, and my experience and our experience here in the lab, like as with everyone with the manual patch technique, of course, you're happy if you get away with a handful of measurements from a day, and so there's always a lot of gunk on those membranes, and often it's really difficult to get the seal and then get your current of interest. We haven't really seen any difference in the difficulty, as it were, on the manual side. It's always a good challenge, whether they're young or old, for us.
And that certainly comes back to almost what you were talking about in your talk, Cadler, in terms of, part of the progress and development you've made in terms of preparing the cells for recording is to potentially get rid of, some of these debris, extracellular matrix that may have formed in long-term cultures. So I guess that's also a potential factor in terms of recording metrics and success rates.
Definitely.
And okay, so I think we answered this one, but I'll just double check with you, with Cadler. Are the action potentials only spontaneous, or did you stimulate your cardiomyocytes?
They were mostly stimulated. We would see 1 or 2 sites per Q plate where we have also spontaneous action potentials. But the majority I showed you, at least today, was stimulated.
And Jung1 asks, when you measure action potentials using APC, do you run on X mode or single hole mode?
So far, we have done only single hole mode, so it's a single cell. But it's worth a try once we get the success rates up to try also multi-hole. With the success rates we have right now, I think it will be a very noisy recording because we had 1 in 10 sites where you would have a cell, but potentially interesting, especially to compare the sum of these different types of action potentials to the single cell measurements.
Yeah, I was just thinking that too, as in, obviously when you move from that sort of single hole to a multi-hole, you may start to like somewhat even out that heterogeneity, that variability that you show so nicely in like the 10 different action potentials that you showed in that figure. So I guess in some ways it kind of gives you a nicer, more even averaged current, but then you do lose some of that granularity that you get from the single hole, where that, for instance, action potential 1 versus action potential 10 could be very different. There could be a whole spectrum of shapes of the action potential in those 10 recordings.
Yeah. What we do right now is, or what would be interesting to start doing now, is to compare the single cell data to monolayer measurements. So you have the combined tissue behavior, and I think the multi-hole data would then come somewhere in between, because you have the cells separated, so they're not interacting anymore. But it would be interesting to see how it compares.
And final 2 questions. So if anyone's got any more questions, you've got to get them in quick. I know we've gone over time, so thanks for sticking around. But if there are any more questions, I know I can stick around. Vil probably has far more interesting things to be doing. Cadler's got a young family to be looking after, but I can always stick around and give you some nonsense answers for a while the experts take off and do more interesting things. Anyway, 2 final questions, unless more coming in the meantime. Is there a way to sort out the different cardiomyocyte subtypes before recording by using a cell sorter? like the Miltenyi that you system that you did use, Cadla?
Yeah, I think there's these markers for atrial and ventricular cells, for example, that you could use and try and sort them out before. So that is definitely a good option.
And finally, the burning question that all people always want to ask: did you use fluoride containing intracellular solution? And that can be both manual or automated, obviously.
For the automated data I showed today, we did not use fluoride. It was physiological.
And of course, so was mine in the manual.
Yeah. And of course, you know, as much as fluoride is useful to improve the seal capabilities, it will affect your cardiac action potential quite significantly. So it's probably far too compromised a situation to be using fluoride in these, what you're hoping is more physiological cardiomyocyte action potential recordings.
You need to be sure of what you're using it for, at least. For some things, I think you can use it if you want to look at a specific ion channel in voltage clamp and test mutants or something like that, and you just want the success rates up. It's okay, but you need to be aware.
Yeah, I agree, and I think that's probably it. And we've probably taken more of people's time than was necessary, but thanks for everyone who stuck around, particularly to the presenters for some great data, great talks, and for staying around so long to answer all the interesting questions too. Thanks to you both. And that concludes it really. And we look forward to the next webinar, which I believe is coming up in probably about a month or so time. So please check our, I don't know, websites, our LinkedIn feeds for future webinars. Till then, take care everyone. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye-bye.